Hello, here is my narration along with a slide deck on all my thesis ideas, updates till now, and the bigger experimental picture. Let's begin. My thesis revolves around how complexity and shape in the morphogenesis leads to complexity in functionality, and can we use morphology as the variable that can explain the differences in the expression profile of the cell, but also the functional circuit formation. And to put it simply, it means that if you have a much more complex structure, as we have in vivo, which goes through different transitory morphogenetic processes, does that lead to much complex or much more in vivo like cytoarchitecture? And also, does that lead to, hence when you have much more complex cytoarchitecture, does that also lead to functional maturity or not? And this is a graphical abstract from one of the papers in which they made two types of brain organoids. One was much more complex, which was much similar to in vivo, and another one which was not embedded in matrixel, which it was not as complex. And then people found out that the one which had more complex morphology, the temporal patterning of the fate was conserved, whereas when you scramble the cytoarchitecture, cells do not know anymore when to differentiate into what. So, to maintain time, we also need to maintain space. Okay. So I have two central questions that I want to test. One is when the self-assembly is to raise the question that if self-assembly is partially encoded by the genes, or and by parts, spatial patterning and morphogenesis plays a role in encoding. In encoding the cytoarchitecture and arrangement of developing brain. So this dogma revolves around that if form leads to function or not, and if we change the form, can we also perturb the local cellular structure, and hence we can also change the functional maturity, whereby changing the functionality of the tissue. Because brain cells, they are not only arranged in a particular manner, but the arrangement itself leads to one particular type of circuit that leads to certain functionality over the other. Okay, so here the spatial configuration is the key, and if you have much more closer in vivo like state, then I'm expecting to have earlier maturity in functionality or activity, and to have better cytoarchitecture. One can also argue another dogma which revolves around that if, let's say, that assembly is purely encoded by the genes, cells themselves know how to connect to one another and form certain temporal they know when to differentiate into what and whom to connect to, then we can change the form, or if we change the form, if we can get similar self-assembly, and that would tell us that self-assembly itself can make robust patterns no matter what your initial form is, and the structure and then hence circuitry is somehow conserved. Okay. And if so, then one can also ask that, okay, let me say that you may not have spatial arrangement, but you have structural maturity, as we have seen in capillary microfluidic project from Sergey in our lab, that structural maturity is reached when you have what you provide much more nutrition to the growing tissue in vitro. So one can ask that in that case you don't have structural arrangement that is preserved as in vivo, but you have structural maturity, and there you may not have as much of neighborhood effect in vivo. And then one can ask that how does the functional maturity begins when you just have structural maturity and no spatial information. Okay, so first we are talking about form and function, and I want to study this in developing human brain. And to know that, we need to know how the human brain forms in the first place in vivo. Okay, so at the level of form, human brain starts from neural tube around 4 to 5 post-conception week, and around 8 conception week, you have vesicles formed, and then they expand, and from telencephalon you have early cortical structures that are formed, and then it gets convoluted to have gyri and sulci in the adult mature cortex. I am limiting my study to telencephalon or forebrain early development in order to understand cortical structures as they arise from rostral end of the neural tube. Okay, so this is at the level of form. What happens at the level of structure or cellular arrangement? That around 4 to 8 post-conception week, you have radial glia cells, neuroectoderm cells, which becomes radial glia cells, and in human specific condition, you have outer radial glia cells that are divided, that are differentiated from radial glia cells. And then first neuronal type emerges, which is Cajal-Retzius cells, and then this is transitory population of neurons. They appear at subplate and then they die off. Then you have intermediate progenitors arising from outer radial glia, and then you have neuroblasts. These neuroblasts migrate radially using radial glia cells to climb up to developing cortical plate, and then differentiate terminally to excitatory and inhibitory neuron starting around 12 to 15 post-conception week. Okay. And at the level of function, I wanted to understand what is the earliest time point in humans available that literature has shown that you have functional activity in these cells as they are developing, but early stage human brain data is mostly absent. People start recording from fetal cortical slices only from gestational week 20. So we are going to use rodent and xenopus information to better understand what type of functionality and what's the role of this functionality as the structure is laid out step by step. Okay. So few examples for human brain data. At gestational week 20 from occipital lobe of human embryo, embryo cortex, this study has shown that subplate neurons, when you patch them, they show action potentials. And repetitive firing, which is a signature of much comparatively mature neurons around subplate. So this scenario is around gestation week 20, and just to remind you that at gestation week 20 you already have so many different types of cells appearing. And earlier than this, what's happening at the level of function is still unknown. Okay, let's move on to next example from human. So this is not functional data, but they have marked the genes for functionality, and also they have recorded from gestational week 26 and also 24, but there was no electrical activity around gestational week 24 prefrontal cortex slices. So they have put the data from gestational week 26 from upper layer, deeper layers, and subplate neurons. And we can see that with they show some sort of activity when you patch them around 50 volts. They have a negative sodium current, and this is different. This is different for different layers of neurons. So there is already some sort of functional segregation that has been happening. And potassium current also increases with your patch voltage. Okay. So from here we can gather the information that around gestational week 26, slices, cortical slices or fetal brain is active. These cells, as soon as they emerge at upper layer, deeper layer, and subplate, they are mature. And also excitatory neurons, they start emerging already around gestation week 8 to 12, and yeah, it keeps on increasing as time goes by. That's it. This shows that structurally or cell fate acquisition is happening over time, and also functionality has been reached around gestational week 26. But we don't know what happens earlier in earlier phases, or what happens if you what is the role of this arrangement that radial glia they have to arrange radially, or neurons have to migrate radially. And if they don't migrate, if they just stay in the deeper layer, then maybe they may not show repetitive action potentials, or they may not show signatures of electrical maturity. Okay, so these were the two major findings from human fetal data. And now we move on to as early as possible embryonic cortex data. And this study from Rush, they have found that as early as embryonic stage, embryonic day 11, if you record from whole embryo, you will see GCAMP. If you label them with GCAMP, you can see calcium activity in cortical cells. At this time, they are BFP positive neuroepithelial cells in the cortical wall. And as we can see that the cells, they are active around this region but not around this region, so we don't know if mostly cells are populated. If this is the representative image of this, we can see that cells are also here, but they have still not acquired the functionality. Okay, and we can see single time series of these calcium potential calcium dynamics to see what is the peak feature of each calcium spike. And just to navigate you around what is happening really around this time is that neural tube starts to close in rodents around embryonic day 8.5, and they fully close it fully closes around E10, and neurogenesis immediately starts after from E10 to E11. And by E15.5, you have already radial glia cells populated in cortical wall, and they make this scaffold from ventricular zone to marginal zone, and neurons are now ready to start migration. Okay, so the same study showed a recording from embryonic day 15.5 from the cortical slices, and you can see that the radial glia cells, they are mostly populated around marginal zone, and they show bidirectional calcium spikes or calcium transients, and they have also shown that if you increase the if you add FGF2, which is a morphogen for brain expansion, you increase the number of calcium events and also duration of them. And they have also shown that calcium transients act as the source of information transfer in the columns, in the protocolumns, as they are forming between the radial glia cells and early neuronal cells to guide them to reach their destinations. And this result that calcium activity helps in or functionality helps in attaining the structure, and it helps in neurogenesis and migration, which further matures the structural maturity of the cortex is quite interesting, and it proves our it's in the same direction as we want to know the role of functionality on the form and the form on functionality, the two-way process. So we are getting in the right direction, at least in terms of what is happening in rodents. Next, if we see, okay, you tell me that radial glia are in, as soon as neural tube is closed and neurogenesis is happening, we know that of course these neurons are going to fire, and it's still a bit later stage, but if you ask that what is happening during neural plate is becoming a neural tube. So in xenopus embryos, they are divided in stages. Around stage 15, the neural plate cells, they show two types of activity. One is spike and one is wave. Waves are much longer, and spikes are much higher in terms of their fluorescence intensity, but much shorter in terms of their durations. And at this time, if this paper shows that if you dissociate the neural plate cells, and only 3% of the cells are active, or they have called only 3% of the cells are spinal cord neurons, early spinal cord neurons. And they have kept them for 6 to 8 hours in vitro culture, and after that they have shown that these cells are electrically active. And when you shift from neural plate to neural tube around stage 19, and if you record this data was from dissociated cell, but if you record from intact neural tube, you can also see spontaneous calcium transients, and the number on these segmented cell indicates the number of times they have shown the calcium transients. And this shows that neural tube cells they show some calcium activity. We don't know if it's related to voltage dynamics or not, or what is happening to the resting membrane potential of these cells. Are these cells capable of any sodium dependent action potentials or not, or is it just calcium that is there at this point, and it is helping in other morphogenetic events. Okay, so to briefly touch upon what is the role of this functionality as the structure is taking shape. I have come across a lot of studies, but the major key takeaways is that with time, as the structure is being attained, there seems to be a mutual relationship between function and form. And we know that the self assembly cannot happen just by genes, and there has to be more along with the transcription factor. There has to be a communication mechanism, and we are hypothesizing that communication mechanism is early stage functional activity that, along with morphogens or in response to morphogens, leads to attainment of mature structure. So researchers have argued that galvanotropism, which is attainment of membrane potentials and electric fields, precedes it precedes the time point when the cells start to respond to chemical cues. So, if one of the research papers have shown that if you experimentally hyperpolarize the neural stem cells, then you can speed up the neural fate. But you can also, if you depolarize the neural stem cells, you can also reverse the differentiated neural stem cells. So this briefly touches upon that with time, apical cells at the ventricular zone, they are constantly being hyperpolarized, and as we know, neurons are stable at hyperpolarized state, and intermediate progenitor is also increasing with time. So intermediate progenitor are produced when there has to be a threshold, when your precursor cells are they attain certain membrane potential. And also with time, radial glia scaffold always remains. They differentiate and divide into different cell types. Cell types migrate, they stay there, they attain their functional maturity, but radial glia itself, they stay in ventricular zone, and with time, ventricular the radial glia, they also start hyperpolarizing, and there has to be a signature of radial glia that has to make intermediate progenitors early on, and its membrane potential versus the radial glia that is at the later stages, and that has to make that has to do gliogenesis. So the fate determination can be linked with its resting membrane potential as a signal to divide into one type of cell type over the other. Okay, so I'm touching very shortly on what can be the mechanisms that link the chemical cues to the um, activity of the cell. By activity, I mean certain resting membrane potential, or some certain ion channels, or ion channels opening and closing, and ion influx and efflux. Mostly calcium at this point, that causes the change in the resting membrane potential. So there are two key findings. One is from stage 19, very early neural tube in xenopus embryos. This study has found that if you increase BMP concentration, then the number of cells that are going to spike calcium transients, they are going to reduce, and it's different. The trends are different in different parts of the neural tube. It's different in the dorsal, whereas it's different in the ventral part. Second study found out that neurotransmitters very early on, for example glutamate, when it binds to NMDA receptors, they can act as morphogens. They allow calcium to get in, which leads to morphogenetic changes, or changing in the shape of the tissue, and leading to flat neural plate to fold into a tube. And differential expression of the glutamate receptors, NMDA receptors, can lead to differences in patterning, which region will fold and which region will stay flat. Okay, so now we need to prove, we already know that how our brain develops, in terms of form, in terms of its structure, in terms of functionality, from limited data points. And from rodents and xenopus, we also know that very early on, as the structure is forming, you have cells that are attaining functionality. Now, can we say that if you remove the structural or form component, can you also have cells being functional? And if you forget about, we know anything about in vitro neuronal culture and recordings from them, it's a very naive question to ask and say that if I don't know who is with me, can I even connect to them and start firing? So it is true that 2D in vitro cultures, they indeed fire. So this study uses iPSC derived neurons, but in very simple, no morphogens added media, only B27 media, and they compared it with human primary neuronal cultures. And you can already see the neurons that have been taken out from the environment, from a very complex environment like human brain, and the de novo cultured neurons, they have structural differences, yes, but they also have functional differences. As you can see from here, that at day 50, the 55, these iPSC derived cultures, they show much less calcium transient numbers as compared to primary cultures, which are much more in number, much frequent, and much complex in terms of number of frequencies that are involved, and spontaneous activity is also higher. This is at day 50 and this is at day 55. Okay, and they show different dynamics, the two cultures, and they indeed the IPC derived culture has higher variability at later stages, whereas human primary cultures, they have attained a certain range of dynamics they are going to follow. So this tells me two things. One is that when you have much more complex organization in vivo, you have much more uniform differentiation of the cells, and hence they would have certain characteristic which is in a very narrow range comparatively to iPSC derived neurons in terms of their functional behavior. Okay. Whereas when you take out that spatial information and you just use iPSC derived cultures, then you can see that cells are still not sure of what to become functionally. Okay. And one may ask, does this is this related to structural maturity or is it related to cell fate specification? Then later on, people found out that if we add certain molecules to inhibit a SMART pathway, then we can get cortical neurons which are much more differentiated in terms as compared to just early stage cortical neurons, and they start showing activity only at day 16. And so this says that even though you disrupt the temporal order, still you can get the function. But still, these electrophysiological signatures, they resemble immature neurons in vivo. Okay. So we have till now asked that how in 2D systems, indeed cells become functionally mature, indeed cells have some cell fate specification, they lack structure. But if we introduce structure, does it take us more to in vivo like functionality, or does it take us to more complexity in terms of how cells are differentiated? So to answer that, we'll go through different protocols and different organoid research. They have who have shown both the transcriptional changes and also the functional changes. Okay, so first is from Kian forebrain organoids. And they have shown that if you compare the organoid age in different regions, in the different hmm, yeah, in the different brain regions and also different ages of the fetal brain, and uh, if you match it or do Pearson correlation with the organoid transcription profile, you'll see that early on the data matches that with early stage fetal brain. Later, in later stages, it gets more and more similar to that of early stage, later stage fetal brain data, and in at around 100 days, the transcription profile is much similar to that of around 24 post-conception week, or in this region maybe 16 to 24 post-conception week, which shows that transcriptionally we are able to add 3D complexity. We are able to add complexity by introducing third dimension to the culture. And also around the 84, there is cortical layering. They shows different cortical layer specific neurons. Not only about the self fate attainment, they also show that extracell excitatory post synaptic potentials, they also become higher. The number of cells that there is, they show excitatory postsynaptic currents, they are much higher as we go in time, and it drops around 100 to 120, maybe because of necrotic core formation or reasons that I don't know. Second is Lancaster type brain organoids, and they are much less guided. They are unguided type of brain organoids, and they're also encapsulated in matrigel. And we can see that they also increases with the functionality also increases with time. Number of spikes increases, number of bursts increases, spike rate increases. But so we can know that of course they are becoming functionally mature, but do they resemble what stage of fetal brain functionally do they resemble, or can I even get to better functionality if I even further improve my in vitro model? Okay, so another study from same Lancaster protocol, they have shown that the spike activity is there at around 7 to 8 months old brain organoids, and they have not compared it with any region or time point in vivo. They have just mentioned that several studies in neuroscience for decades, they know that if you plot counts over ISI interspike interval, you're going to see this distribution. And this distribution, if you have these parameters, this distribution, this is variance, this is mean, this is coefficient of variation, and this distribution, also known as Poissonian distribution, if you get this distribution, that means that you have neuronal data. So they show that indeed we are able to get functional maturity, but still, when these protocols generate 3D organoids, are they skipping some cells? Are they reaching cell fates, and are they reaching to mature neurons without getting to getting through immature states, or how the transition is happening is quite interesting. Okay, so now another study. This is semi-guided protocol, and they made cortical organoids from iPSC derived iPSCs, and they were able to show complex neural activity at 8 months, and the oscillatory activity increased over time. And they also compared organoids with just 2D culture using the same protocol, and they found out that organoid has again lesser variation as compared to 2D, and mean firing rate also increases over time. We don't know what happens after this or if they have discontinued the culture, but in this region itself we can see the trends are very different. So from here we can say that indeed structural complexity has to do something for neuronal communication to happen in much more human way. And they compared the preterm neonatal data from 24 week to 38 week of postmenstrual age, and they found out that only the organoids in the later stages matches to that data, and neither the mouse pyramidal primary culture, nor the 2D iPSC derived culture, nor the early fetal cells, and also the organoids in the earlier stages, they are able to correlate to the EEG patterns that was seen in fetal brain. Okay, so from here we gathered this information that humans are incredibly complex, yes, but you cannot have any type of geometry or topology and differentiate cells to get to what functionally happens in the brain. You also need neighbors to chat to. You also need a scaffold to shorten the distance between neuron to neuron, and all these features combinatorially affect the maturity of the developing brain. Okay, so this study from Tanaka in 2020, they compared all different types of brain organoid protocols with that of fetal brain, and the trajectory that is followed from neuroectodermal cells to neurons to astrocytes and other type of cells, they have tried to see if the organoids also follow the same trajectory or not. Some of the protocols, they show that they also have non-specific cells, non-specific differentiation, but more or less they follow the same trajectory. I have picked up the Trujillo protocol to move forward with, because they have shown that semi-guided differentiation can lead to more complex neural oscillations, and they have much more similarity to the fetal brain as compared to unguided protocols, and also they have less non-specific cells that are differentiating. Okay. So, also some proofs on comparing now 2D with 3D systems electrophysiologically, and also, as I mentioned earlier, that if you keep even the 2D, but over time you gain the structural maturity, you have the cells terminally differentiated, what happens to their electrophysiological activity? And from first study, we can see that 2D cells versus 3D neurospheres, there is indeed a lot of differences in their electrophysiological properties. And even the 2D cells, as they acquire the fate over time, they start from no action potential to abortive action potential to repetitive action potential. And we can see that indeed cell fate is there, that's playing the component in the functional maturity. Genes are essential, but shape is essential as well. Okay, so we are not the first one to get this idea. This has already been tested out not too long ago, only in 2023, in canonical Lancaster type brain organoids, that if you do not embed them in Matrigel, um, and And if you embed them in Matrigel, how differentially you'll get the cells that are the amount of cells, the spatial distribution of cells, and much more mature cells. And from here we can see that the simple cytoarchitecture has much less variability in terms of cells, and much more complex cytoarchitecture shows spatial arrangement of different types of cells and much more number of cells, much more types of cells. This is spatial transcriptomics of cerebral brain organoids from Lancaster type protocol. And not only that, if you plot them in latent time space, you can see that when you have no complex structure, cells forget when do they have to develop into what. So if you have complex cytoarchitecture, you can also see that the cells, they follow temporal order, as we see from ventricular zone to marginal zone. And this is very crucial because as you have much more complex cytoarchitecture, much more preserved temporal information, you'll see much more human in vivo like features as compared to when you don't have cellular cytoarchitecture complexity. So this tells us that brain organoids, they have, if you want them to mimic in vivo better, you not only need morphology that is complex, but also you need timing that is accurate. And cells also need spatial coordinates to undergo correct temporal progression. So when they lose the position in space, they also lose the maturation timing. This ties very closely to what we are trying to find in our hypothesis. But the difference is that we want to further improve our in vitro cultures and tie the component of functionality to it. So coming down to two competing dogmas of ours, we need to do certain tests to prove if indeed higher complexity in structure and form leads to higher maturation rate, and order has to be preserved in terms of functionality. And if I see that if you have more in vivo like data, transcriptomic data, then your activity level also is enhanced. Or our second dogma that structural maturity is enough and neighborhood has no effect on it. So to do that, I am yet to make a very, very, very detailed experimental plan. But here is the overview of what I will try to target for different testing different dogmas. So first, we need yet another in in vitro model, which is not assembling through aggregation process, but a um, tubular tube closure as it happens in vivo. That by this we are not only changing the complexity, but we are also changing the initial conditions. So if you have two different initial conditions, and if they come to similar local global minima, or if you have different initial conditions, you're going to get different cellular architecture. So first, I will start with different self-assembly processes. One is aggregation and another is tube closure. We already have model for this, and I have to culture them for longer terms in order to record activity from them. And then I need to map cell fate determination of these two models of self-assembly using semi-guided protocol, Trujillo protocol, along with a component that will maintain the cellular structure, that is hydrogel embeddings. And once we have a map of cell fate specification in both these systems, I want to record functional maturity by imaging calcium dynamics and membrane voltage dynamics, because early on, calcium is the common denominator for a lot of different processes, and I would not know if this process is relating to any sort of membrane voltage changes or bioelectric potentials. And along with not only dynamical changes of functional maturity, I want to measure the markers that lead to functional maturity. For example, ion channels, voltage gated ion channels, or even certain neurotransmitter or neurotransmitter receptors. We can try tying the cell fate specification and functional maturity along with time in these two different models. And once the functional dynamics are compared, we will read the structural organization. The cell fate determination and structural organization are more or less similar. That if we use spatial transcriptomics data or using lineage tracing, either we can use omics database lineage tracing, or we can tag a nucleus with a certain promoter and fluorescent protein, and then we can see how it divides over time. Or we can also use CRISPR based barcoding methods of the cell to study where in space cells are dividing and which cells show what type of functional activity. And then I would like to perturb, so we have to measure players, functionality and form, and I would like to perturb one by one each one of them by keeping the other constant. So first I would like to perturb functionality by optogenetics. So I would like to play with inducing functionality, and then I would see what happens to the structural arrangement. Is my structural arrangement improve? Do I see more migration of neurons? Do I see much more neurogenesis, or can I shunt the neurogenesis altogether and say that in later on in closed loop, functionality is also affected by stimulating and perturbing the structural formation. And then next player I would like to perturb is cytoarchitecture. I would try to play with the cytoarchitecture. One, we are already changing the self-assembly initial stages by having two different self-assembly processes. So, but later on we can further perturb cytoarchitecture by eliminating embedding or by introducing stretching, and by changing some morphogens in our protocol, and then reading out what happens to my spontaneous functionality emergence. And this is for our dogma number one or hypothesis number one, that you need both spatial, temporal information in order to get to correct functionality. Then, second, to test second dogma, I would say that I don't need self-assembly at all. I'm going to provide cells to gain structural maturity as they would gain in vivo, but I would not help them to make very nice cytoarchitecture as it happens in vivo. So I would like to do this with our capillary microfluidics platform, and I'll compare that with the tube model to see what happens to the function. That indeed my capillary model, as shown in Sergey's paper, that it shows better functional markers, much more mature functional markers, but also it doesn't have the spatial arrangement. So what is happening to it as compared to the tube? And even after keeping everything in vivo, like in the tubes, we are trying to make them matrigel tubes, we are embedding them, we are trying to help them make single lumen and let them be there, let these single tubes be there for longer term. And still, if they form multiple lumens, then we can ask the question that what it is in the genetics of the cell, that even when I'm changing the spatial information, the cells are arranging themselves in multiple lumens, and something has to be there still in the spatial arrangement that causes the multiple lumens to form and arise within the well-controlled tubes. I am still working on very specific experimental plan along with the along with time dimension in my PhD, and I might come to it very soon. But to come to the first point, that we need a better in vitro model, which are tubes. They would be much more in vivo like, they would have one single lumen, and then we can think about functionality. And what I have tried so far regarding generating better tubes. So I have done multiple trials. So just to make everyone aware that at day 8 we have a tube inside a tube model. If you add BMP4, you will get this beautiful tube inside a tube. And I have done this with different cell lines, mainly GCamp and COLF, and I can see that they show very different structures at day 8 itself. And GCamp shows a very thin tube in the middle, whereas COLF shows a very thick tube, and the space between neuroectoderm and ectoderm is very small, whereas it's not the case in GCamp. Then I tried to grow them for long term by doing peg embeddings and doing just with GCAM to record also the calcium activity. And in the initial trials, I couldn't keep the tube in a tube alive for even at day 11. And there was no activity that was seen. Next trial, I was able to keep them till 14 days, same tube in a tube, but no peg embeddings. And around 14 day, it flattened out and cells started to die, and no flushes were observed. Then I did next trial with, I thought the peg embedding is almost crucial, and then kept them till day 26. Still no flashes were observed, but, and also the cells, the tube shrunk progressively, and it was no longer there at day 26. Next trial was with adding 6% matrigel, and we found out that with 6% matrigel there is something happening to the lumen formation, because I was having problems in initial matrigel lumen formation, especially in GCAP. But I still couldn't keep them for long term, even till day 9 or day 10, because they start to shrink very quickly and die. And I tried Trujillo protocol for the first time, and I saw that with 6% matrigel, these organoids, even at day 4, it form better lumens. This is day 4 image, and we can see it has a better lumen. And I was very happy, but it never reproduced again. It was never reproduced again. At later stages, I saw that they became very huge. They increased in size, and even before I could embed them, they detached. So I can try embedding them earlier before they detach next time. But coming to next trial, I tried both Casbrun and Trujillo protocol with both BMP and no BMP, and also with 3 cell types, and also played with embeddings. I'm showing the key conditions. Other conditions, they died off even quickly. So when you have COLF and with BMP, around day 46, they lose their tubular structure, and if you do not add BMP, then there is some structure, and but I don't know what is happening, because there were a lot of cell death around the tissue. And around when you have Trujillo and embedding tubes for COLF, it was very pretty luminized structure, and this lumen was way longer than it happens in self-organizing lumens. But it later detached, because Trujillo organoids, they are getting very thick very quickly. And coming to wild type sigma cell, I was able to replicate Dries and Elena's protocol and keep them for 51 days, but later on they also I couldn't fix them because of fungal infection. But I tried wild type sigma with Trujillo and it didn't respond, whereas COLF, which is another wild type cell line, it responded well. So I don't know what's going on. I tried yet another time GCAM with BMP, and I saw the similar result, that tube shrunk and died. And also when you don't add BMP, you see in all different cell lines that when you don't have BMP, you have a lot of cellular migration out of the tube, and cells, they come off very easily from the pattern. The GCAM Trujillo with embedding, they survived for day 53, even though I saw in my calcium videos, I saw random luminized structure formation. Because they do not form single tube early on, maybe that's why they are behaving as a solid cylinder, because they are solid cylinders, and they are behaving as aggregates and forming multiple lumens. But I was indeed able to keep them till day 53 and saw some some calcium activity. This is my most latest experiment, and I was able to keep them till day 23 because of fungal infection. I had to abort them, and I used two cell line Colf and GCAP. And this time I didn't use the with BMP condition because it was not staying for very long. And at day 21, Colf without BMP, and all of them were embedded, and they were also compared to aggregates. When I don't add BMP, I'm seeing several lumens formation, which is similar to Elena's and Reese's results. And Trujillo, indeed, again, some luminized structure formation. In aggregates Colf around the same day, I see the same lumen structure, but huge, with huge core. Or I don't know if it's one single lumen because it seems like a solid a ball. GCAMs at day 21, they again showed a luminized structure, very pretty structure, and with Trujillo, and with again, multiple lumens are self-organizing themselves. At day 21, GCAM without BMP, again the same cell death is happening. They are detaching a lot, so I have to figure out a way what to do with them being detached. And GCAM Trujillo aggregates look way different than Colf, and they do not form this outer neuroepithelial structure. So I don't know what is happening. For the next steps, I would focus on first and very quickly model building. I would try to maintain single lumen. I would try to, because Trujillo protocol doesn't give me initial matrigel responded lumens, I would try to make lumens from the Carsburn and then switch to Trujillo for the expansion. And then I'm developing the calcium and membrane potential tools where cell line is in development. And further, after meetings and discussions, I'll come up to time targeted experimental plan. That's it. Thank you.